Most brain health protocols treat food as background, the things you should generally eat more or less of, while the real cognitive intervention work is done by supplements. Today, we are making the case that 4 specific whole foods deserve to be treated as direct neurological interventions, as precisely as any supplement in the protocol. Not because eating well is generally good for brain health, though it is, but because these 4 specific foods contain specific compounds with documented mechanisms that no supplement replicates. Compounds that reach brain tissue through unique pathways, that accumulate in the specific brain regions where cognitive aging is most active, that activate biological mechanisms, blood-brain barrier crossing, gut fermentation products, direct cerebral blood flow, that make their brain health effects categorically distinct from their supplement analogs. The first is wild blueberries. Not regular blueberries, wild blueberries, which contain dramatically higher anthocyanin concentrations than cultivated varieties. Research found that wild blueberry anthocyanins cross the blood-brain barrier and accumulate specifically in the hippocampus, the memory center most vulnerable to cognitive aging. In the hippocampus, they directly activate the BDNF signaling pathway through a mechanism that involves both direct receptor activation and reduction of neuroinflammation. The second is pomegranate. Pomegranate's elagitannins, the polyphenols responsible for its color, are converted by specific gut bacteria into urolithin A, the mitophagy activator. But food-derived urolithin A, produced through gut fermentation, has metabolic characteristics that differ from supplemental versions, making regular pomegranate consumption a valuable complement. The third is dark chocolate. The flavanol compounds in high cocoa dark chocolate produce a measurable increase in cerebral blood flow within 2 hours of consumption. This happens through a nitric oxide-dependent endothelial mechanism. A second mechanism, BDNF elevation, has been documented specifically in the hippocampus. The fourth is strawberries, the richest whole food source of fisetin, the natural senolytic we've established in the senolytic protocol. Regular strawberry consumption provides ongoing background fisetin intake that contributes to the clearance of senescent cells, complementing the periodic higher-dose supplemental fisetin pulses. By the end of this video, you will have a specific dose-calibrated food protocol for each of these 4 foods and a framework for your daily diet. The wild blueberry research is among the most compelling food-brain science in the aging literature, and the distinction from cultivated blueberries deserves detailed explanation. Wild blueberries have significantly higher anthocyanin concentrations than cultivated high-bush blueberries. Research from the USDA found that wild blueberries contain 2 to 3 times the anthocyanin content per gram as typical cultivated varieties. This concentration difference matters because brain health effects appear to be dose-dependent, with wild varieties producing more robust effects. Wild blueberries contain a broader spectrum of anthocyanin subtypes, including delphinidin and malvidin, while cultivated blueberries contain a more limited spectrum. Research suggests that these specific combinations cross the blood-brain barrier more efficiently than the narrower spectrum of cultivated varieties. Wild blueberry anthocyanins specifically accumulate in hippocampal tissue, a finding documented using radioactive labeled tracers to follow their distribution. This region is most critical for memory and most vulnerable to aging, making wild blueberries specifically relevant to cognitive health. In the hippocampus, these anthocyanins activate BDNF through 2 mechanisms: direct promoter activation and suppression of neuroinflammation. Both mechanisms work together to produce the BDNF elevation that current research documents for brain protection. Clinical trials confirm this. A study in the Annals of Neurology found significant memory improvements in adults over 16 weeks of supplementation. Plasma anthocyanin levels predicted the magnitude of improvement, confirming the dose-dependent mechanism behind these cognitive gains. Recommended intake is 1 cup daily, fresh, frozen, or freeze-dried. Frozen varieties are widely available and retain their content effectively. Freeze-dried powder provides a concentrated form that can be easily added to smoothies or your morning preparations. Pomegranate and urolithin A. Pomegranate's elagitinins, primarily punicalagin and its metabolites, are the most extensively studied dietary precursors to urolithin A. When consumed and metabolized by specific gut bacteria, particularly Gordonobacter urolithin faciens, elagitanins are converted to urolithin A and related urolithin compounds. The important distinction between dietary and supplemental urolithin A involves the gut fermentation step. When urolithin A precursors are consumed as whole food, the urolithin is produced in the gut, where it can interact with the gut epithelium. This may produce distinct local gut effects, including direct gut microbiome modulation and gut immune activation, that purified supplemental urolithin A does not replicate. Research has found that pomegranate juice consumption significantly increases fecal urolithin A output, confirming meaningful gut conversion. Daily intake. Half a pomegranate or 120 milliliters of pure pomegranate juice provides meaningful elagitannin content for urolithin A production. The fermentation efficiency varies between individuals depending on gut microbiome composition, which is why precision probiotic protocols amplify the brain health benefits. Dark chocolate and cerebral blood flow. The flavanol research on dark chocolate, specifically adipokine, has produced dramatically visible brain health effects. Research used brain MRI to measure cerebral blood flow, finding a measurable 2-hour improvement following consumption of high flavanol dark chocolate. Adipokine directly activates ENOS in brain blood vessel endothelial cells, producing nitric oxide-driven vasodilation that increases cerebral blood flow. The research-supported intake, 30 to 40 grams of dark chocolate with 70 to 85% cocoa content daily. Milk chocolate blocks flavanol absorption through a milk protein binding mechanism and should not be substituted. Strawberries and fisetin. Strawberries are by far the richest whole food source of fisetin, containing approximately 160 micrograms per gram fresh weight. Regular strawberry consumption provides ongoing background fisetin levels that contribute to senolytic and anti-inflammatory biology. While concentrations from dietary strawberries are below the threshold for acute senolytic clearance, they provide ongoing background activity that complements supplemental fisetin. Daily intake. 150 to 200 grams of fresh or frozen strawberries provides meaningful fisetin and additional polyphenol content for longevity. One of the most compelling aspects of the food polyphenol research is the synergy between different polyphenol classes, mechanisms through which different compounds in different foods amplify each other's effects. Wild blueberry anthocyanins and dark chocolate flavanols both produce cerebral blood flow improvements through different upstream mechanisms that converge on nitric oxide and vascular regulation. Consumed together, they produce additive cerebral blood flow improvements that neither achieves alone. Pomegranate elagitinins and strawberry fisetin both support senescent cell biology. Pomegranate through urolithin, amatophagy activation. Strawberries through fisetin senolytic activity. Consumed regularly, they address two complementary aspects of the cellular aging biology that the supplemental senolytic protocol addresses, creating dietary reinforcement of the pharmaceutical approach. Dark chocolates adipokine and blueberry anthocyanins both activate the NRF2 antioxidant pathway through different upstream sensor mechanisms that produce additive NRF2 activation when combined. Research has documented synergistic antioxidant gene expression from combined consumption that exceeds the sum of their individual effects. The practical implication is that consuming all four foods regularly produces larger total brain health effects through polyphenol synergy than any individual food alone. The daily polyphenol protocol is designed around this synergy. The complete dietary polyphenol protocol integrates the four brain-building foods into daily eating patterns that are sustainable and practically achievable. Morning, wild blueberries. 1 cup of frozen wild blueberries added to a smoothie, to yogurt, or to oatmeal provides the daily hippocampal anthocyanin dose. Frozen wild blueberries are available in most grocery stores and retain full anthocyanin content. Consuming them with the morning protocol takes no additional preparation time. Important sourcing note. Look specifically for wild blueberries. They are typically smaller and more intensely colored than cultivated blueberries. Mid-morning or midday. Pomegranate. Half a fresh pomegranate or 120 milliliters of unsweetened 100% pomegranate juice consumed with a meal or snack. Look for 100% pomegranate juice without added sugar. Alternatively, pomegranate arrows added to salads provide fiber that supports gut fermentation. Afternoon or evening, dark chocolate. 30 to 40 grams of 70 to 85% cocoa dark chocolate. Consume it standalone rather than with milk to ensure flavanol absorption. Afternoon timing is preferred as the modest caffeine may affect sensitive sleepers. Daily, strawberries. 150 to 200 grams of strawberries, fresh or frozen, consumed at any meal or snack. Strawberries are affordable and widely available year-round. Frozen varieties retain fisetin content comparably to fresh, making them a great staple. This protocol requires no cooking and replaces existing snacks with brain-building alternatives. Simple, sustainable, and powerful for cognitive health. The dietary polyphenol protocol connects to the complete brain health system through the BDNF, cerebral blood flow, mitophagy, senolytic, and antioxidant threads. Wild blueberries and BDNF. The lion's mane and turmeric morning coffee provides the NGF and anti-inflammatory BDNF support. Wild blueberry anthocyanins directly activate the BDNF pathway in the hippocampus through their accumulated presence in hippocampal tissue. The two approaches activate BDNF in the same brain region through complementary mechanisms. One through chemistry, one through antioxidant effects. Pomegranate and mitophagy. Supplemental urolithin A activates mitophagy through the direct compound. Pomegranate consumption provides precursors that the gut microbiome converts to urolithin A through natural fermentation. The two approaches together provide both direct and precursor-based mitophagy support. Dark chocolate and cerebral blood flow. Beetroot extract provides dietary nitrate-driven NO production for cerebral vasodilation. Dark chocolate provides epicatechin-driven eNOS activation for cerebral vasodilation. Two independent mechanisms of cerebral blood flow improvement. Strawberries and senolytic activity. Supplemental fisetin provides the acute senolytic pulse every 6 weeks. Daily strawberry consumption provides background senolytic activity. The dietary and supplemental approaches together provide more continuous coverage. Here is your specific action plan for this week. Add wild blueberries to your morning routine this week. Frozen is fine. Add dark chocolate to your afternoon. Add pomegranate to one daily meal. And incorporate strawberries as a regular snack. These 4 additions require minimal preparation and produce measurable brain health effects that compound with every week of consistent consumption. Next week on Neurovitality After 50, we are going into the specific cognitive impact of spices beyond turmeric. We'll explore saffron, ashwagandha, black seed oil, and herbs like rosemary and sage, each with documented neuroprotective mechanisms. Subscribe now. Turn on notifications. Share this with someone whose diet deserves the brain-building foods it was designed for. Thank you for being here. Stay sharp, stay nourished, stay vital. I will see you next week.